Okay, here we are for the second part of Chapter 7, Part B. We just got done with the lymphatic system, looking at there's vessels, there's primary organs and secondary organs, and now we're going to look at how those interact with the immune system. So the immune system we have been hinting at for chapters 4, 5, 6, and we're finally arrived where we can go into the immune system. Now, I want to give you a heads up. The immune system is complicated. There's no way around it. I will make it as simple as I possibly can, but you do need to plan to just set aside some time, work on this, perhaps make your own concept map. I'm not requiring it, but if you liked that for the blood chapter, give it a whirl with this one. It might help a little bit. And what I find helpful is to focus on smaller bits, and then once you feel really good about the first and second and third line of defense, then start going back, putting it together, asking questions that might overlap just a little bit, and we'll get through it that way. One of the reasons that it does seem complicated to people is that this is the one system where we don't have any organs, like the lungs or the heart or something really big that we can see and touch and hold. It all has to do with cells and proteins and much smaller things. And so you just got to trust me that what I'm saying here is what's happening. There's a lot of information and a lot of research going on in the immune system right now, so it's totally possible that next week or in 2 years or in 20 years you are going to hear something that might be just a little bit different than what you hear today when you listen to this, because we're learning so much more about it every day. But for your exam, what we will use is what's in the book, which is what we know up until this point. But absolutely, I'm excited to find out what else we learn in the next few years and decades. And then, of course, we'll update the information at that time. So for right now, today, we're going to look at what our book has published, which was in the last 3 years, so it's fairly up to date, and we'll go from there. So to actually get to what is on my slide here, we've got in the immune system 3 lines of defense, and they certainly go first, second, third, in that the body will first try to use some things to block a pathogen, and then if that doesn't work, the second line will come into play, and if that doesn't work, the third line will come into play. But do keep in mind that those lines of defense overlap. So if it goes through the first line of defense, the second and the third lines of defense might be activated around the same time, and then so there's some things going on there. But for the most part, we always learn it first, second, third to keep them straight. So your first line of defense is an innate defense. You are born with it. It is innate within you. So therefore, it is non-specific. It doesn't care what type of pathogen it is. Could be a virus, could be a bacteria, could be a piece of dust. It doesn't matter what it is. It's going to try to prevent it from getting in the body. So the first line of defense, you have 2 categories, physical and chemical. We'll go into those in a moment. The second line of defense will come into play if something has breached the first line of defense. So in the second line of defense, something has entered your body. This is also an innate defense. You are born with the second line of defense, and that also means it is non-specific. It does not matter if it is a virus or a bacteria or a prion or something else. It will go after it if it doesn't think that it's part of your body. 3 big categories in the second line of defense. You have white blood cells phagocytizing pathogens, you have inflammation occurring, and you have protective proteins. And again, more on each of those 3 coming in a few slides. Then your third line of defense. You are not born with this, and that's why we call it adaptive, because your body, as it is exposed to more things, adapts to that and generates the third line of defense. So this is very specific. This is going to be specific to one particular type of virus. It's not the same as any other type of virus, much less a bacteria. But if you have a virus, let's say a virus that has ABC as some sort of code, and then you have a virus that's ABD as its code, this third line of defense will only work for one of those. And then you'd have to be exposed to the second one before you can fight off the second one. More on that later, but just remember it is specific, where the first and second are non-specific. And in our third line of defense, we have B cells and T cells, which are the exact same things as B lymphocytes and T lymphocytes. And remember right now that lymphocytes were one of those 5 types of white blood cells that we talked about in the blood chapter. Okay. So, innate immunity, non-specific defenses. In your first line of defense, we want to prevent something from entering the body. Let me get a little color here. So what we want to do is we want to have a barrier to entry. So you guys can immediately look at your body and say, yeah, skin, that's a barrier, right? It's real, all those cells are so close together, and we're waterproof, and they slough off. We've got this thick, stratified squamous layer. That's why we learned about those tissues. Then so we're a little bit protected against cuts. So that's our barrier to entry, is going to be skin. That's going to be a big one. Okay. We also have mucous membranes. Okay. So you might notice, you might be saying, well, Rachel, I've got skin covering my body, but what about my mouth? What about my nose? What about my ears and my eyes and some other openings I have between my legs, right? We have vaginas, we have urethras, we all have, well, probably all of us have an anus unless you've had some surgeries done. So we all have these openings, and those things, they might be lined by skin eventually, but not all the way down. So we do have skin lining many of those openings, at least at the beginning, but then what we have in all of those is a mucous membrane. And that mucous membrane, so if you, let's see, let's draw a rudimentary mouth here. Okay, there's my mouth, and it's got a pharynx coming out the back. Sorry, that should be open, heading down to my esophagus, right? Okay, very rudimentary. So that's what I've got going on here. And lining all of this all along the side are cells that secrete mucus. And mucus, if you've ever blown your nose, you will know is quite sticky. So if I'm a virus and I get drawn in because you're breathing, but I get stuck to the side of your esophagus or your mouth, then what can happen, this is what phlegm is your body trying to cough that up so that you can spit it out, or if it gets into your nose so you can blow it out, and sometimes even your body will swallow it because your stomach acid is meant to destroy it. So that's the benefit of mucous membranes. And that's, you have mucous membranes on these other parts of your body too. You have it lining your anus, you have it lining your urethra a little bit. The vagina tends to be a little bit more, a little bit different, but you've still got mucous membranes along the reproductive tract. And so that's there to catch pathogens if there's an opening. Okay, so we've got skin and mucous membranes. And again, remember, you're born with those things. They are innate, and they don't care what it is. They're going to try to trap it and keep it out of the body. So that's our first line of defense. Our second, let's label that. First is over here doing a rockin' job. But as you know, sometimes we get cuts, or sometimes we get sick and something has gone down our throats, and the mucous membranes have not been able to protect us in that moment. So that's when the second line of defense will come in. Let me change my color. So those are all these arrows. The second line of defense is going to be all of these things here. They are still innate. We basically have our own concept map right here. They're still innate because you're born with them, and they are non-specific. So you have protective proteins If we follow that arrow all the way down, it reaches us here where we see something called complement proteins and interferons. So complement proteins are super great because they are going to basically try to kill pathogens. They just happen to be proteins. They're not whole cells. But if they notice something shouldn't be there, they're going to go after it. Interferons are nice because if a cell becomes infected with a virus, it's going to want to warn its neighbors that that's coming, and so it will produce and release interferons and send them out of its cell so that the neighbor can say, oh, we've got a pathogen in the neighborhood, we've got to put up some barriers so that it doesn't infect me too. So again, they're great because they're proteins, they're not entire cells, they're little, they're itty bitty, but they still protect us. All right, another part of the second line of defense are the phagocytes and the natural killer cells. So, really awesome name, right? Natural killer cells, pretty straightforward. Some people call them NK cells. But basically, these are cells now, not proteins, but whole cells that are out there to phagocytize. That's why we learned that word back in chapter 3, and they're going to phagocytize these pathogens, which in this case are the green oval shapes. And so macrophages can serve as phagocytes. Monocytes are actually going to turn into macrophages. Remember, that was a type of white blood cell. Neutrophils are a type of phagocyte. Dendritic cells might sound familiar from chapter 4. All of those things can serve as phagocytes. And the natural killer cells, of course, too. Then another part of the second line of defense is the inflammatory response, and we'll see more about the inflammatory response later. But essentially, when you think of inflammation, you think red, you think pain, you think heat, and that's because we're going to try to bring a lot of blood to the area because blood carries white blood cells, and all of these things here are white blood cells. So if we can get more of them to that location where there might be an infection, we will be better off at protecting the body. So here is all of that in text, okay? First line of defense. In our physical barriers, we have skin, and we mentioned those mucous membranes. You also have tears and saliva and urine, and those things tend to either be a little bit acidic or they tend to contain different enzymes that go after bacterial walls so that the bacteria can't survive. You also have normal bacteria. This is what we might call your gut flora. This is the bacteria that lives in your gut normally, and when they live in your gut, they're totally taking up all of the room and all of the nutrients in there. And by doing that, they're just out-competing pathogens from becoming established. So, you know, if you imagine you're going down a row of houses and you're looking for a house to buy or a place to live, but all the houses have people living in them, then you're out of luck. In that spot, you just got to keep moving. You're not going to stay there. And that's what's going on, is that we've lined your gut with so many good bacteria that the pathogens have nowhere to stay. Chemical barriers are going to include sebum. We mentioned that, right? Antimicrobial properties in sebum from your sebaceous glands, going all over the skin, out of the hair follicles. You also have lysozyme. That's one of those enzymes in saliva and tears and sweat that goes after bacterial cell walls. And if a bacteria doesn't have a cell wall, it can't survive. So these are going to line not only your skin, but also those openings that we mentioned. And then you have an acidic pH in your stomach, so anything that does happen to get swallowed is fairly likely to be destroyed in the stomach. And you also have an acidic vagina if you're a female, and that is meant, of course, not to harm necessarily the sperm or harm the penis, but it's meant to protect the female body from an infection in the vagina or the cervix or the uterus. All right, so our second line of defense, we said we have protective proteins, we have natural killer cells and some phagocytes, and we have the inflammatory response. So here is that inflammatory response, which is just one part of the second line of defense. So in the inflammatory response, we do kind of want to track what's happening. So what happens is some of your tissue is injured. So basically what we should envision is some sort of knife or something came in here, shoop, and cut your, maybe it was an accident, maybe it was just a pin, but something cut your cells, made a little opening, and now all these pathogens that were outside of the body have finally found a way to make themselves known inside the body. They found an opening. So that's happened. That's in the body. But at the same time, your tissues that were injured from that cut are going to release something called histamine. Okay, we've got probably 3 new chemicals, and I want you to keep track of histamine because that's one of them. So the injured tissue releases histamine, and that histamine causes capillaries to dilate. They get bigger, and that's going to increase blood flow. And what did we say was in blood? Right? White blood cells, those natural killer cells, nutrients, oxygen, things to make those cells make ATP and do their job. So all these things that you need to heal and fight infection are in the blood. So histamine is going to dilate the capillaries, and we increase blood flow. All right. Now, while that is going on, right now we're going to avoid the blood clot. While we've got blood flow increasing, we are going to see macrophages exiting the blood capillaries. Remember we said these are the sneaky teenagers, right? Look at them right here. They're totally sneaking out of those cracks in the capillary wall. And monocytes are one type of white blood cell. Neutrophils are another type of white blood cell. They serve as macrophages, which is just a really generic phagocytizing cell. So those macrophages are going to phagocytize as many pathogens as they can. And here's our second chemical. Remember we said there were going to be 2. Our second chemical are cytokines. Okay, so the macrophages are going to release cytokines, and that's going to stimulate more of the inflammatory response. All right, so more of that inflammatory response. So we kind of have a feedback loop here where more cytokines means more blood, more capillaries dilating, more neutrophils and monocytes squeezing through the capillary wall, phagocytizing pathogens. And so the cytokines just kind of say like, okay, yeah, here's the party, here's where you need to go, this is where we need to have some action. So that's going to be going on in the inflammatory response. And then the last part in this picture here is just pointing out that if a blood vessel itself happened to be damaged in the process of your injury, then of course we would be creating a blood clot at the same time to prevent blood loss. Because while we do want those things in blood in the tissues, if we just have full-on blood, it's going to be too thick and it's not going to be able to heal. So we don't want that. All right, so here are those protective proteins that we mentioned earlier. You have complement proteins and interferons, and they're both just proteins. So that's why compared to this whole green cell, the complement protein is just this tiny little thing right here. So complement really means they complement the immune system cells. They're helping the immune system cells. And so they can do a lot of different things. Someone I know working at the University of Minnesota Duluth, she's looking at one little part of the complement system in pregnant women with hypertension. So she's looking at one little tiny spot. That's her research, and when she knows more, she'll publish it, and more people can build off of that. So there's all these little pieces, but we can kind of put 2 big categories here for complement proteins. They can attract phagocytes. So they can be this kind of red flag saying, hey, 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 the infection's right here, I found it, this is what you need to attack, basically. So they're kind of very helpful that way. The other thing they can do, which is what we see in this particular image, is that they can poke holes in bacterial walls. So they actually come in here, this is kind of a cool video game thing to do, and they all line up in this big circle, they punch holes in the bacterial wall. You can imagine these others come in and they complete this circle, and now you've got this big pore into the bacterial wall. And you can imagine, think back to osmosis, right? We've got all these things inside this bacteria, all these solutes. So water is going to rush into those holes. It's going to rush into the bacteria. And what did we say? If something rushes in, right, it's going to swell and possibly burst. And because these holes are so massive, it's really, really likely that that bacteria will burst and therefore be dead. So that's what complement can do for your body when fighting an infection. The other type of protective protein are interferons. So interferons, we kind of said this, but here's the definition: made by virus-infected cells sent to warn neighboring healthy cells, basically to put up a barrier. So let's imagine if we have a row of cells here. This is just going to be super simplified, okay? And the pathogen so far today has been green, so we'll stick with that. So let's say this one is infected with our pathogen, our green pathogen. Now, that cell is going to know it's infected, but it can't actually do anything about it. Our cells aren't designed, these cells aren't designed that way. Excuse me, not designed, they haven't evolved that way. So because they don't have the ability to fight the infection, what they do have the ability to do is create these things called interferons. And the interferons will leave the cell and head in here to this cell, and they'll leave the cell and head over here to our neighboring cells. And then what that does in the neighboring cells is say, hey, guys, this is a really not good pathogen, and it's already infected your neighbor. So you've got to put up this big barrier. You've got to make some extra protective proteins. You've got to make some barriers and make sure all of your proteins on the outside are being really good bodyguards, and don't let them in. That's what we're stimulating. That's what the interferons are going to stimulate. All right, so those were all innate defenses. Your body had those when you were born. The things that you have been developing and are continuing to develop to this day are your adaptive immune defenses. This is your third line of defense. And the third line of defense is really helping protect the body against two things: specific pathogens, very often after your non-specific defenses have failed. And the second thing it helps protect against is cancer and cancerous cells. And it does this essentially the same way. So for the most part, I'll just be talking about pathogens some more, but it does apply to cancerous cells. Now, as we know, we can all get sick from something to the point that we die, depending on it, and the same is true of cancer. So your body is always going to be trying to fight these, but sometimes the pathogen or the cancer cells are just too strong and overpower it. So that's why we've got to do our part, get our checkups, keep doing our self-checkups at home, trying to avoid exposure to carcinogens when we know what they are, right? We know that smoke is one of them. We know that asbestos is one. There's whole lists of these on the internet if you're not sure of what they are. So we've got to do our part, but unfortunately sometimes it does overcome our own body's defenses. So here's what we're hoping happens as we fight off infections. Your B cells and your T cells are basically going to be doing all the work. What you're going to discover is there are different types of B cells and T cells. So we'll see that in a moment, but they're basically doing all of the muscle work for the adaptive immune defenses. So, antibody-mediated immunity by B cells. It's a heck of a name. I know it is. I did not name it. Antibody-mediated immunity. Okay. The big word, what's going to change is this one, antibody. When we talk about T cells, it's something else. When we talk about B cells, it's always going to be antibody-mediated immunity. So what's happening is that when you think back to blood, red blood cells, do you remember, I hope you remember, that red blood cells have antigens on them. They have what is now looking like these little Y things sticking out of them. But other pathogens also have antigens on them. And sometimes, actually many times, you just have antigens floating around in your body, not actually attached to cells. So we're kind of expanding on that idea. So activation is the first part of this process. It is when a B cell receptor binds to an antigen, activation occurs. So in our particular image that we're seeing here, we have an antigen, and we're assuming at this point that's foreign. That's not, it's not part of your body. And this particular antigen, if we have to look at it and we had to decide if that was circular or V-shaped or square shape, I think we would probably all notice that that's fairly circular. Okay, so it's going to fit into this circular. Now I've covered it, you can't really see it, but if you look at these other ones right here, these other B cell receptors, they are circular, versus these B cell receptors are triangular, and these are a boxy sort of shape. So what's going to happen is that only, I'm going to put a star on it, only this B cell is activated. Okay, that's the one that becomes activated because that's the one that bound or was able to bind to the antigen. So now when we look at this next part, look over here to the second part of this. The B cell that was activated is going to divide into plasma cells and memory B cells. Okay, so just right now, the activated B cell will divide into plasma cells and memory B cells. Two types of B cells. All right, so let's read the caption here. Clonal expansion, basically making copies of yourself, right? Making clones. During clonal expansion, cytokines secreted by helper T cells stimulate B cells to clone mostly into plasma cells or memory cells. That's basically a big fancy way of saying what I just said, right? B cell receptors, or the B cell receptor binds, the B cell divides into plasma cells and memory B cells. And something that had to happen to make that happen was we needed a cytokine that came from a helper T cell. Okay, we'll learn about helper T cells in a moment, but they were just helping. Okay, so if we could put together, so we've got plasma cells, we've got memory B cells, and what you might notice, if you look at all of these receptors now on all of these memory B cells, they're all those circular ones. In this image, there isn't a single triangular or boxy receptor. They are all circular because those are being stimulated. The plasma cells then also are able to produce these receptors with circles, but they get released. So this is what we call an antibody. Well, you can see antibodies right here, okay? This little Y-shaped thing is an antibody, and the green thing that it's bound to is an antigen. All right, so that's why we call it antibody-mediated immunity. The plasma cells are just going to pump out antibodies. They make tons and tons and tons. We're talking like thousands a second. They make tons of antibodies. Okay, so those antibodies, they're going out. The plasma cells are just shooting them up, bing, 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 and the antibodies are rushing through the blood, and they're getting into the tissues, and as soon as they find those antigens, they bind them up, and they say, haha, I caught you, you're not going anywhere, and then the antibodies can actually clump together, and then they just kind of float around, and now a white blood cell, maybe a macrophage, maybe a neutrophil, can come along and say like, oh hey, I recognize you because you're this massive clump of antigen and it was really easy to find you because you were all clumped together, and then it's able to destroy it. So that is super helpful, and it was the antibodies released from the plasma cells that did all of that work. Now, the plasma cells themselves and the antibodies themselves cannot actually destroy an antigen. Plasma cells can only make antibodies. Antibodies can only bind to antigens. But in doing those steps, that allows a cytotoxic T cell or a neutrophil or something else to actually then do the destroying part of the job. So, very, very helpful. Memory B cells, they're just going to hang out for the rest of your life in your body, waiting, and in case they encounter that antigen again, then they're like, hey, I know what to do, and I can do it very fast because I'm already ready to go. And we're working on it that way. Okay, so once your infection has cleared, once your body has been able to get that infection taken care of it, it's gone, then we have something called apoptosis. And that is programmed cell death. Some people call it cellular suicide. Basically, our plasma cells, now that we don't need all these antibodies anymore, are just going to go through a process of taking in water and bursting. Okay, so that was all with B cells. It was antibody-mediated immunity, and now we're going to look at a second part of the third line of defense, a second part of adaptive immunity, and that is your T cells. And you have 3 types of T cells in your body: cytotoxic T cells, helper T cells, and memory T cells. Now, you can use this little T with the subscript C if that helps you to shorten it, no problem. So cytotoxic T cells, they are pretty awesome. Remember, cyto means cell, and toxic, you guys can probably figure that out, means toxic. So cytotoxic T cells do not contain toxins, but they are absolutely toxic or deadly to other cells like pathogens that are infected your body. So they have vacuoles. Remember vacuoles? We talked about them a little bit in chapter 3. And they have granzymes in them and perforins in them. Now, I know we've thrown out a lot of words, so let's review. There is something called lysozyme in your tears and your saliva. There is something called histamine released from injured tissue. There's something called cytokines, which are released to enhance inflammation. And then we have granzymes and perforins. So granzymes are, well, let's do perforins first. So perforins, just like thinking that you perforate something, right? It has holes in it. Perforins are going to punch holes into a cell. So when we look at this top picture here, the perforins are these orange things. Very, very similar, you might notice, to those complement proteins we talked about earlier. It's just that these proteins happen to be coming from outside of a cytotoxic, or from inside a cytotoxic T cell. Once the perforins have essentially made a perforation, they've made a hole, then the granzymes, which were inside, can enter that pathogenic cell, and it can get inside and force apoptosis. It can kind of get into the DNA. Well, it could do it a number of ways, but it could then force that pathogenic cell to go through apoptosis. So remember, that would lead to cell death, which means it was toxic to the cell. The cytotoxic T cell did all of that. All right, then we have helper T cells, which is just like the name sounds like, they are helpful. So they release cytokines to help your B cells divide. We mentioned that on the previous slide, but here's the connection. They release cytokines and they help the B cells divide into those plasma cells and memory B cells. Technically, they do a lot of other things, but for our class, that's all we need to know. The third type of T cells are memory T cells. And it's the same idea as memory B cells, it's just that they're T cells. So memory T cells are going to be able to kill basically in the future, or they're going to be able to recognize and attack a pathogen in the future much, much faster than your body was able to the first time. And this is what's going to give you immunity. This is how, when you got the chicken pox, I know some of you, I guess, got the vaccine for it, but it worked the same way. You know, I got the chicken pox from my sister, and my body, in response to that, made a ton of memory T cells. So now anytime I'm exposed to that particular virus, and because the chicken pox virus does not mutate very often, when I'm exposed to it, my memory T cells, even if it gets in my body, my memory T cells can go right to it, latch on, and divide so quickly that they're able to fight off that infection so fast that I never actually get sick from it. So, super awesome. So here is, those were the 3 types of T cells. This is a lot of text, I know, to just help us put that into an order. So this is cell-mediated immunity by T cells as opposed to the antibody-mediated immunity by B cells. And remember, we called it antibody-mediated because antibodies were doing something. And here we're calling it cell-mediated because cells are going to do something. So what's happening? We have a macrophage. It phagocytizes a pathogen. Okay, that's what we've got going on right here. Okay, so the macrophage phagocytizes a pathogen, breaks it down, and displays part of the pathogen's antigen on the outside. So this itty-bitty green thing that you see right here that I'm now essentially covering up in black, that was this little part right here of this pathogen. But now it's displayed on the outside. And at that point, we call this macrophage, we no longer call it a macrophage. We call it an antigen presenting cell. This becomes an APC because it is literally presenting an antigen on the outside. It's as if, this is kind of gruesome, but if, you know, in cultures where you might display, let's say you got in a fight with a bear and then you won, and the bear was now dead, you could then take its teeth and wear it as a necklace, or its claws or something. That would be you presenting the thing that you had tackled and conquered. That is what's happening here, is we're taking a piece of the bacterium or a piece of the pathogen and presenting it on the outside. And now our antigen presenting cell is just going to make its way to the lymph node or to the spleen. Remember, those were our secondary lymphatic organs. Okay, so we've got our antigen presenting cell. It's made it to, let's say, the spleen, and in that spleen we have a generic T cell that is able to bind to that antigen. And it's going to use the same thing. You know, you can see here it still has these antibodies sort of extending, but that's how it recognizes it, or a receptor, a T cell receptor that recognizes that antigen. Okay, so we have recognition, and in response to that recognition, we have activation. So that T cell is activated into a cytotoxic T cell. And now that cytotoxic T cell is able to divide. It's able to make helper T cells, divide into helper T cells. It's able to divide into cytotoxic T cells and memory T cells. And all of those cells divide. Divide. That's our clonal expansion, right? We're just making lots and lots of clones of the same cell so that we have all of the same receptors. And now the cytotoxic T cells, they all have this receptor for this antigen. So now they can go out into your blood. They'll leave the spleen. They'll go back in the blood. They'll escape. Remember, they'll do the sneaky teenager. They'll escape through the capillaries, head out to that damaged tissue. And as soon as they see that pathogen, now remember, they can see this whole pathogen now because all they need to do is recognize this little part in the corner. And as soon as they recognize that part, they'll use their perforins and their granzymes to kill those infected cells. So they could go after the pathogen directly, or perhaps an infected cell that's also displaying an antigen. It could be either one. The T helper cells are going to help signal the B cells to get on a move with their antibodies. And when the infection is removed, then we do the same thing that plasma cells did. The cytotoxic T cells and the helper T cells are going to go through apoptosis because we don't need them anymore. And your memory T cells are going to continue to hang out in your body to protect you from any future exposure to that same pathogen. So, I know that was a lot. I hope that was clear. Please review anything you need to. I do recommend making yourself a concept map. Send me an email because there is a bit there, but I have full faith that you can sort it all out.